Okay, we're back. It's really awkward recording myself, so I have to have some fun with it. We're going to be doing the structure of a nephron, which is the basic functional unit of the kidney. So as I talked about earlier, we have the renal arteries, which are going to go into interlobar, and then arcuate, and then interlobular. Now we're into the afferent, so that's going to be those smaller branches of the interlobular. So that's going to bring our blood into the glomerulus, which is going to be a bundle of arterioles. And then after it leaves and is filtered at the site, then it's going to go away in the efferent. So that's the same with our nerves. We have afferent bringing it towards, efferent leaving. So glomerulus, bundle of arterioles. These are going to be really small vessels. And then we have the Bowman's capsule, which is going to run around. And so the glomerulus and the Bowman's capsule, or the renal corpuscle, this is going to be where filtration takes place. So we're getting everything out of our blood that we don't want, and that's going to be filtered at this site. Does that through a lot of those chemistry processes that we discussed earlier in the cell biology chapter, a lot of diffusion, concentration gradients. All of that stuff is going to be incorporated here in that area, but you'll get more into that in lecture. We're going to do pretty basic here in lab. So after we have this blood filtering component, glomerulus, and the Bowman's capsule that kind of surrounds that bundle of arterioles, we are then going to go into the proximal convoluted tubule. So that's going to be kind of a random squirrely little thing. So proximal meaning closer, convoluted tubule. And then we are going to go into the descending and then ascending. So descending going down, ascending coming up. And this right here is the loop of Henle. So we have a lot of reabsorption happening. So sometimes when we filter things out, there's still things that we want in that filtrate for our body. So that could be water, that could be different ions. So we're going to reabsorb it back into our blood throughout these parts. So descending and ascending. And a lot of this material is covered on different entrance exams for professional schools. So really important just to know the basic structure and functions of different parts of the nephron. So we have descending, ascending on our loop of Henle. So descending tubule, ascending tubule. And then we're going to go into the distal, so distal meaning farther away, convoluted tubule. I'm almost running out of space, so I'm going to tilt this a little bit. Okay. And then after this, we have reabsorbed everything back into our body that we want out of the filtrate. And then we've put all the stuff that we don't want into our filtrate that's being carried. And so now this can kind of be filtered out into collecting ducts. So you can have multiple nephrons putting all their stuff, dumping all of their filtrate into the collecting duct. Then these collecting ducts are going to run down into our calyces, and that's going to let our urine eventually go into our ureters and into our bladder. So that is the basic structure.